Everyone, welcome to this Cerebellar Ataxia research update as this year's virtual conference. It's great to see so many people here, including some familiar names that I recognise. But for anyone that doesn't know me, I'm Emily Cutting. I'm the research manager here at Ataxia UK. I'll talk now for about 20 or 25 minutes, and then we should have some time for questions. So if you hold on to them till the end, I'll put them in the chat. We'll get to them. And then next is the doctor's Q&A session. So I'll start with an overview of what I'll be talking about. I'll start with Ataxia UK research activities. I'll talk about developments in diagnosis of ataxia, in understanding the progression of ataxia, in developments in finding treatments, and I'll finish by covering some opportunities to take part in research. And I'll keep coming back to this overview slide, so hopefully everyone can follow along with the different topics. So starting with Ataxia UK activities in the world of research. Some people here, I think, will know a lot about what the research team does as Ataxia UK, but others might not know so much. So I've just got a couple of slides about the main ways that we at Ataxia UK support research. So probably the most obvious way is that we fund research projects, and this is made possible by generous donations and contributions from our fundraising community. We fund a very broad spectrum of research, from very early stage projects to contributing towards clinical trials, and we also cover the full range of ataxias. Every year we ask our funded researchers to tell us the ways our funding has impacted their work. And from this, we know that every £1 Ataxia UK spends on research projects raises £5 in further funding for ataxia research. And this shows that the relatively small grants that we give allow researchers to gather important data, which enables them to apply for bigger grants from elsewhere. We also arrange research events like the International Congress for Ataxia Research. This is done in collaboration with two American charities, NAF and FARA, and the Ataxia Global Initiative. ICAR 2024 in London was the biggest conference yet, with 80 talks and 350 research posters. Conferences like this allow researchers to come together to discuss their work, and they're a really good catalyst for moving research forward, so they're really important. And other events include our educational webinars, like the gene therapy webinar that some of you may have attended, that was a few years ago now. We also work as the link between researchers and the ataxia community. In this role, we help recruit participants to research studies, and we facilitate including the patient voice in research. We work with pharmaceutical and biotech companies to encourage them to develop treatments for the ataxias. And I think you'll see over the course of this talk that interest from pharma companies is growing. We assist with their drug development programs in any way we can. This involves anything from introducing companies to neurologists that are interested in running clinical trials, to collecting information from people with ataxia using surveys or interviews. This blue box here shows an example of a project we worked on with Biohaven, which looked at the burden of living with different scars from the perspective of the patient. And this is vital information for pharma companies to direct their research, but also when it comes to applying for regulatory approval for treatments, this is really important information. And all is to say that we are engaged in all stages of research. Finally on this, there's a few other aims of the research team, which I won't cover in detail, but if anyone is particularly interested in the work of the research team, you can read all about our aims in our research strategy, which is available on our website. So we aim to put the results of research into practice. We involve people with ataxia in research and represent people with ataxia in international initiatives. And we also aim to publicise ataxia research as widely as we can. So now I'll move on to the main topics for this research update, starting with developments in diagnosis of ataxia. And here I'll cover new genes that cause ataxia, NHS genetic testing, and diagnosis of non-inherited ataxias. So inherited ataxias are caused by changes to our genes, which are then passed down through the family. And I'm sure a lot of people here are familiar with that concept. New genetic causes of ataxia are being identified all the time. And we've been talking about this one for a while now, but in 2019, the genetic cause of canvas was identified, and that is the mutation in the RFC1 gene. And in 2022, a genetic ataxia called SCAR27B was discovered, which is caused by a mutation in the FGF14 gene. And both of these are late onset ataxias, and SCAR27B can start with episodes of ataxia symptoms. A treatment's being tested for SCAR27B, which I'll talk about more a bit later. And the important thing to know here is that if you don't have a specific diagnosis for your ataxia, and by this I mean a cause for your ataxia hasn't been identified, which is quite common amongst people with ataxia, then you could speak to your neurologist about being tested for these conditions. They both appear to be more common causes of ataxia, so a lot of people are being diagnosed with them. And we have information on our website about how to access these tests and how to access specialist ataxia centres. If you have any problems with this, you can contact the research team or our helpline. On the topic of genetic testing, we recently published an article in our magazine about why it could be worth being tested again if you haven't had genetic tests for a number of years. And there are two main reasons for this. The first that I just described is that new forms of genetic ataxia have been identified in recent years, which makes getting a diagnosis more likely. And the second reason is that genetic testing has changed dramatically in recent years. The technology used has advanced a lot. So a new technology available on the NHS called whole genome sequencing means that hundreds of genes can be checked simultaneously rather than one at a time. So if you have ataxia with an unknown cause and you haven't had genetic testing for many years, then you could speak to your GP or neurologist about being tested again. So moving on to immune-mediated ataxias. And over the last few years, there have been changes in the diagnosis of immune-mediated ataxias as well. And there are different types of these ataxias. They all start with a trigger. The trigger initiates an immune response, which is like the body reacting to the trigger as if it's harmful. So an immune response begins, and for some reason, this immune response attacks the cerebellum. And damage to the cerebellum is what causes the ataxia. We know of a few different triggers that can cause immune-mediated ataxia. Gluten can be a trigger, and gluten ataxia is treatable with a gluten-free diet. And sometimes the trigger is unknown, and in these cases, the condition is known as PACA. And the team at the Sheffield Ataxia Centre have published research papers on these types of ataxia, including how to diagnose them and how they can potentially be treated. This information is on our website. You can email the research team if you're interested in learning more about this. We're co-funding a research project at the Sheffield Ataxia Centre, which is designed to improve diagnosis of gluten ataxia, and I'll speak more about that towards the end of the presentation. Moving on to understanding the progression of ataxia. Understanding the progression of ataxia is really important. It's important for people living with ataxia so we can better understand how the condition progresses and what to expect. But it's also really important for clinical trials and for developing treatments. And there are a number of active research projects which study the progression of different ataxias, and they're generally called natural history studies. And some people here might be involved in one of these already. So I've got some examples here. The CRC SCAR looks at scars in the US, with Ready SCAR looking at SCAR1 and SCAR3. ESME is a SCAR3 natural history study, which includes a site in London, and there's a worldwide DRPLA natural history study. And the PROSPECTS project looks at the progression of RSACs and SPG7. And there's lots of other rarer ataxias being studied in smaller research projects. And the Ataxia Global Initiative is a worldwide network of ataxia specialists with the goal to facilitate development of treatments for ataxia. The aim of the AGI is all about understanding the progression of different ataxias and the best way to measure progression for trials. Our DRPLA research manager, Sylvia Prades, has a role on the AGI in the executive committee, and that comes back to our aim of representing people with ataxia in international initiatives. So just to highlight a couple of things that AGI is working on, which is shown in bold here, they've published a number of papers on standardizing data and sample collection. And this means neurologists taking samples for research are doing it all the same way and taking the same samples so that the data set that can be used in research is as useful and as uniform as possible. They're also looking at development of outcome assessments to use in trials. So this is the thing that's measured in trials, so researchers can tell if the treatment being tested is working. For a progressive condition like ataxia, which can progress quite slowly, this is a really difficult thing to do. The AGI are looking at the SARA score, which you may all be familiar with as a measure of ataxia symptoms, and they're looking at making sure that changes to the SARA score actually reflect how the patient feels when going about their life. And this is important so we know that trials are measuring something that's meaningful to people living with the condition. And it's also something that regulators are interested in. So the AGI is a very busy network, and we are starting to see results coming from this group now, which is very exciting. To finish this section, I've got a couple of Ataxia UK funded research projects looking at progression of different ataxias. And these projects were funded recently, so they're still in the early days. We funded a SCAR27B project looking at which other genes might impact the age that SCAR27B symptoms start and how the condition progresses. So although, as I said earlier, the genetic cause of SCAR27B has been identified, researchers believe that other genes could have an effect on progression. And this project will look at the genes of 1,000 people with SCAR27B to see which other genes could be involved. As well as helping us to understand the condition, this could identify new targets for treatment. We also recently funded a SCAR3 and Canvas study in Australia, and these researchers will look at health data from people with SCAR3 and Canvas to see if they can identify patterns within these groups, again, to learn about other factors which could be influencing the progression of these forms of ataxia. So on to the next topic, which is developments in finding treatments for ataxia. This section is probably the longest one, and will cover genetic-based therapies, other treatments in clinical trials, and research aimed at supporting people affected by ataxia. So I'll start by talking about genetic-based therapies, which have potential in treating inherited or genetic ataxias. So this includes gene therapy, gene editing, and antisense oligonucleotides. And don't worry if you've never heard any of those terms before. I just want to show here that there are different types of genetic-based therapies, and all of these approaches are being studied for the ataxias. And some of these studies are starting to enter clinical trials. So Arrowhead Pharmaceuticals are starting a trial testing a SCAR2 treatment. SCAR2 is caused by a genetic change which results in the production of an unhealthy ataxin 2 protein, and their treatment aims to prevent the production of the ataxin 2 protein. This trial will include sites in London and Newcastle. We don't yet know when the recruitment for that will start. Cure Rare Disease received a $5 million grant to advance their SCAR3 antisense oligonucleotide program. And an N of 1 trial has started in the US testing an antisense oligonucleotide for DRPLA, which is a rare form of ataxia. There are other genetic treatments in development for other ataxias, but this is just a few examples that I wanted to pick out. Ataxia UK funded a project looking at genetic treatment for SCAR3. This is preclinical work, so it's currently being tested in cells in the lab. People with SCAR3 inherit the SCAR3 causing gene from one parent and a healthy version of the gene from the other parent. And this healthy version has a role in the body. So the healthy version ideally needs to be allowed to function. And these researchers in Germany are looking at a genetic treatment to target only the SCAR3 causing version of the gene, allowing the healthy gene to function as it's supposed to. And like I said, this is preclinical work, so we look forward to hearing the results of this project and seeing how we can support the researchers with this moving forward. So moving away from genetic therapies and into other treatment approaches. Lots of you will have heard about the Biohaven trial before. We've been following this for some years. Biohaven is a company, and they have a drug called troriluzole, which is a potential symptom relief treatment for a number of different scars. They announced positive results from their trial around this time last year. The trial took place over 3 years, and it compared the progression of people on the trial to natural history data. So these are the studies I talked about earlier, looking at progression of ataxia over time. And having this data allowed them to make the comparison. So that shows the importance of these types of studies. Following their clinical trial, Biohaven applied to the FDA, which is the drug regulator in the US, and they're waiting for a response to that application, and that's expected around the end of this year. Unfortunately, in March of this year, they withdrew from the EMA regulatory process, which is the regulators in the EU. The reason for this was the EMA said there was insufficient evidence to support approval. Biohaven have told us and told the ataxia community that they're committed to finding a path forward for troriluzole. So while this is very disappointing, we are in contact with them and will provide any support we can to help with this process. So moving on to a different ataxia and a different trial. Celaxa are carrying out a phase 3 trial testing a treatment for SCAR27B. I mentioned earlier the genetic cause of SCAR27B was identified recently, and it appears to be a more common form of ataxia. Soon after SCAR27B was identified, a small study was carried out to test whether a treatment called 4AP would be effective. And this small study showed that 6 out of 7 people taking 4AP reported a response to the treatment. And Celaxa are now testing a modified form of 4AP in a phase 3 trial in the US. And we'll report results of this as soon as we know them in all our usual communication channels. IntraBio is another company we've been talking about for some time now. They have a drug approved in the US by the FDA called Acnersa. And this drug is approved for the treatment of NPC, which is a condition that has ataxia as a symptom. It's a symptom relief treatment, so they want to study whether it could be effective for people with other ataxias. They're planning a trial in CACNA1A related ataxias, which means ataxias caused by a change in the CACNA1A gene. And this includes SCAR6 and episodic ataxia type 2 and some other rare conditions. They plan to start this trial in 2026 and are currently exploring UK sites. Moving on to some Ataxia UK funded research. We recently funded a project by Dr. Zamy at Aston University, who's looking at augmented reality as a treatment for people with ataxia. Augmented reality is a bit like virtual reality, but it adds computer-generated images to the real world. So people see an exaggerated version of their own movements. So what Dr. Zamy wants to know is whether this can help the brain make adjustments to improve stability in people with ataxia. Participants for this study will need to visit Aston University twice a week for 8 to 12 weeks with some other assessment visits too. And we have some information on our website on the inclusion and exclusion criteria. So if you're interested in taking part, that would be the best place to start, and you could see if you would be able to be involved. And if you're interested, you can email Dr. Zamy to let him know.
Has Emily frozen?
Study people with diagnosis can be referred to Sheffield to be tested for gluten ataxia. Those that are diagnosed with gluten ataxia in this study will then be followed up after 1 year to assess the impact of a gluten-free diet. And that's the treatment for gluten ataxia. And this is a collaborative project with involvement from a number of other hospitals in the UK. If anyone's interested in taking part in this study, you can see our website for more information and ask your neurologist for a referral. So moving on to something slightly different, Ataxia UK's mission, which you might be aware of, is to find a cure for ataxia and to support everyone affected by ataxia until a cure is found. So not all the research we fund is aimed at finding treatments for the ataxias, but can also be research which is carried out to support people affected by ataxia in other ways. An example of this is a project by Maria Kearney and Professor Lowett, who's shown in that picture. They're designing audio support messages for people with ataxia. So some people here might be familiar with the I'm Not Drunk card, and Professor Lowett likened this to a telephone version of the I'm Not Drunk card. So it's an audio message that can be used on the phone to explain why your speech might not be there. And Professor Lowett is looking for input from people with ataxia for this project. So keep an eye on our communication channels for ways to be involved in that. And another project that Professor Lowett is working on is focused on people that care for people with ataxia. As I'm sure everyone here appreciates, carers need support too. And Professor Lowett is looking to develop support mechanisms and a support package for carers. And again, she's looking for input from people with ataxia and their carers in this project. Scarlett, our research communications officer, is compiling a list of people that are interested in being involved. So if you'd like to be added to this list, you can email Scarlett. And finally, I've just got one slide on some other opportunities to take part in research. I've already mentioned lots of trials and studies that some people here might be able to take part in. So just to finish off on a few others. The DRPLA Natural History Study is recruiting people with DRPLA and people without ataxia. And the ESME study is recruiting people with SCAR3 for a natural history study. There's the RPROMS project, which is looking at remote assessment of ataxia and whether this can be used to measure progression. And there's a survey for people with SCAR6 and SCAR27B, which is aiming to understand the genetic and environmental factors that can influence these ataxias. And finally, the gene therapy survey, which we've publicised a lot over the last few months, so a lot of people have probably completed it already. And this is a survey designed to understand attitudes towards gene therapy, and it's specifically for people with genetic ataxias. So I've given lots of information there, particularly about different studies that are ongoing. So if you want any more information on any of these, you can find it here on our website. You go to Ataxia Research at the top, taking part in research, and then you'll see the section for people with cerebellar ataxia. Of course, you can email the research team with any questions. So thank you, everybody, for listening.